Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My goal here is to find the exceptional individuals in their fields, interview them, and ask them good questions so they hopefully even say that that's a good question, and then pass that knowledge on to listeners. So my guest today is Curtis Suttle. He's a professor at the Institute for Oceans and Fisheries, the Department of Earth, Ocean, and Atmospheric Sciences. We're going to talk about how viruses work in the oceans and what their goal is, and all the things that go on with them. So Curtis, thanks for coming. How you doing?
Doing very well, thanks. Nice to meet you.
Yeah. So how did you get interested in viruses and the environment?
Well, I guess it depends how far back you want to go. I mean, if we go way back, I was, as a child, even sail around a sailboat.
Oh, wow.
When I was 12 years old, and we were sailing up a research vessel anchored there, had set up, and they were seeing all of marine life, being saltwater crocodile. As I went through and went to what I was really interested, what regulates population. It's quite striking. So what we see in a few milliliters of seawater for hundreds of other questions, how do all these exist? Well, there was that. And so for my PhD work, I'm very interested in what regulates these populations and looking at a variety of factors. And my PhD work was actually done here at the University of British Columbia. And then I went off and did postdoctoral work at Stony Brook University in the United States with a fellow called Jed Furman. And he worked on bacteria in the ocean. And I was working on these small single-celled plants, which we call phytoplankton, which is the basis of the food web in the ocean. But at about that time, we're going back a while now, we're going back into the late 80s, was this sudden realization that, in fact, that bacteria were really what's most important in the oceans. Just to put that into perspective, most people don't think about it, but if you were to take all the living material in the ocean and could weigh it, and on one side of that balance, you put everything that you're most familiar with, anything you could see, basically, whether it be fish or seaweed or limpets or whales, or put everything on one side of the balance. And then the other side of the balance, you put everything you couldn't see because it was microscopic. It turns out that about probably more than 95% of the living material in the oceans by weight is microscopic. And to put that into perspective, those microbes produce about half the oxygen on the planet. And they also turn over very fast. They also grow very quickly. And so I was interested in taking some of my ideas from my PhD and this postdoctoral work, trying to understand what was regulating these microbial populations in the ocean. And at that time, I was working with actually the important role that nutrients. And while I was in that lab, there was a graduate student named Lita Proctor who was working with Jed Furman as a postdoc in it. And what she had found as she was looking under the microscope, she found all of these viruses. I mean, just amazing amounts of viruses. And prior to that, people had kind of talked about why there weren't any viruses and why you wouldn't expect to find them. And so when I left Stony Brook University and took a faculty position at the University of Texas at that time, I decided, well, this sounds like probably the most important thing that regulates populations in the ocean. They're probably viruses being obligate. So we did some experiments looking at whether viruses might infect phytoplankton. Lita Proctor's work and Jed's work was focused on bacteria, the heterotrophic, things that use organic carbon, and I was interested in things that do photosynthesis. So, and the first experiment that we did was very successful and showed that viruses actually were important factors in killing phytoplankton in the ocean. And so this is how we actually got started in it. So my background is actually ecology and population ecology.
Well, a couple of questions here. You said in the beginning you grew up on a sailboat. You sailed around the world. What was that like? Before we get more back into the science, what was it like to live that life? What did you guys do every day? And did you, go on land all the time, or you stay in the boat? What was that like?
Yeah, I mean, it was actually quite incredible. We were the first Canadian family actually to sail around the world and on a sailboat, and it was 4 years. And I left when I was 9 years old and came back when I was 14. It was just my mother, my stepfather, and myself. Stepfather built the boat. It's not like we were wealthy. And if you go back at that time, it's pretty hard to imagine. We didn't have any refrigeration, electricity. There was no digital watches or clocks. We actually navigated the same way that Christopher Columbus did, using a sextant and shooting the stars, and a gimbaled clock. We tried to, no radio. And then even at 9 years old, I had 8 hours of watches I had to keep a day, 2 4-hour blocks, three of us. From 9 years old, I was steering and sailing this boat essentially by myself. With help, my parents needed it, but they had to. It was an amazing experience and certainly shaped my life.
Yeah, that's amazing. What was it like at various points around the world? It seems like, sadly, today, I don't even know if you'd be allowed to do it, or maybe international law would leave you alone. But what did you guys do when how often were you near land versus just out in the ocean?
Yeah, I mean, it really is quite remarkable. Again, to throw you back in time when we did that, there was places that we sailed into that declared national holidays because we sailed into port, right? And there was places that we were the first kind of sailboat yacht we'll call it a yacht. It was only 36 feet long, but that we went into. So we were the first sailboats, for example, to day sail the Brazilian coast. So we would go into a little different port every day, and all the villages would come out and take a look at us so we were very much an anomaly. And so, and there were so few people doing it at that time that you actually knew all the other boats in the world that were sailing in the world and where they were at the mouth, what little island they were in. So, it was quite a remarkable way to grow up. Of course, we had big storms, and there was, and my stepfather, again, would stop for periods of time to work because, again, my parents sold our house in order to be able to finance the voyage, and my stepdad built the boat. But so we stopped in Hawaii for 6 months, for example, and then also we stayed for 1 year in South Africa, where he also worked to build up some savings.
You weren't afraid of pirates or anything? Were they prevalent back then?
Yes, they were. I mean, we were at certain parts of the world and areas that are still quite troublesome, but maybe not as much as they were then. So, for example, the really scariest parts were off the northern coast of Australia by Timor at that time, which is now all part of Indonesia. That was we heard lots of stories of people that had been overrun and killed. And again, when you're in a sailboat that has a small auxiliary motor that can do only about 10 miles an hour full out, you are worried about those sorts of things. And off the coast of northern coast of South America, again, was an area that we had some trouble, and it was quite scary. So, yeah. So not what you would consider sort of a typical upbringing. And I don't think any of my parents had even graduated from high school. So the fact that I would go off and chase an academic career was certainly that was in the cards for them.
Yeah. Have you ever repeated at least parts of the journey with a modern craft recently?
No, not at all. I mean, as an oceanographer, we go out on ships sometimes, but no, I've never. Again, once you start to chase an academic life, even if you kind of wanted to do those sorts of things, you just don't have that kind of time. I did a lot of sailing for a while, like mostly dinghy sailing, and I do racing, race small boats and race bigger yachts as well. But again, not transoceanic racing.
Last question about this aspect of your life, but were there any parts, I mean, you couldn't go everywhere, but I'm sure there were big stretches of ocean that you didn't go through. And are there parts of the world that really no one goes through or sails through?
Even now? I really don't know. I mean, actually, I think people go pretty much everywhere now in the most extreme places. And even back then, there was these folks that people would talk about, just the kind of crazy people that would go down to Antarctica, and there was books written about 3 times around the Horn, and there was always people that kind of pushed the limits in terms of where they went to, and it was an unusual time because, again, not many people were doing that, and so people would show up in island where they hadn't people like coming like this before. But there's a pattern that people tend to follow because of the wind. So the prevailing winds. So I'm sure most people have heard of the trade winds, for example. And so most of the sailboats, they'll always go from east to west because that's the prevailing wind. So it's kind of the same directions that the sailing ships used to follow in the old days.
Okay, I got it. Well then, so fast forwarding, you were talking about how you're interested in bacteria and then viruses based on what had been discovered. So what, as a quick summary, and we'll go into more detail, what is the dynamic overall in the oceans? What are viruses doing? What are bacteria doing? I guess they're nutrient cycling. Like, what do you see?
Yeah, so this is the thing that's kind of remarkable and kind of represents, I guess, a paradigm shift in the way we looked at things because as I mentioned, that the oceans are responsible for about half the oxygen production on the Earth, just to give one example. And yet the lifespan of the organisms that are doing all of this heavy lifting for the planet, photosynthesis, is only about maybe about 8 days or 4 days. It's kind of that kind of range. And it turns out that now we know that viruses are responsible for killing about 20% of the living material by weight in the ocean every day. So the wheels in the ocean spin incredibly fast. And so there's all kinds of implications of that. And because things, the cycles are so quick and the implications are so large when you think of things like the balance of carbon dioxide in the atmosphere versus the ocean. And it's ultimately the microbes that are driving much of this, right? Because they're the photosynthetic organisms that are using up the CO2, for example, using oxygen. And then the other thing that's really interesting, again, if you think about regulating populations, viruses tend to be incredibly specific, right? So we've got this terrible situation with the new coronavirus, which is circulating around right now, and which has obviously jumped from an organism into the human population. And viruses do that, but it's rare. And so mostly the viruses that infect an organism infect only that organism, and often only even a strain of that. And so viruses have this really important role that of maintaining this balance of different species, because when a species becomes too abundant, then viruses are able to propagate quite effectively through that species, and it tends to knock down that population. And so there's some phytoplankton, for example, that form these massive blooms that you can see from space, and then all of a sudden they're gone. And that was, again, one of the things that drove me to thinking about viruses, because there was all this anecdotal literature that was out there. People talked about giant blooms of phytoplankton disappearing overnight. There was reports in the literature of people looking at phytoplankton under electron microscopy and seeing virus particles. And so there was...
so you think that a bloom happens, it's a huge concentration of a certain plankton, and then viruses have a field day and just attack it all?
Exactly. And so we know that some of the biggest blooms of phytoplankton in the ocean now are actually regulated by viral infections. So it turns out that they are... that's exactly what they're doing. And it's complex because, again, what viruses see as a species or a taxon is not necessarily the same thing that we perceive, because, again, viruses have to be able to infect a host, which have to be able to get in. And so, again, going back to the coronavirus that's causing COVID, it had to have a very specific receptor molecule that allows it to get into human cells. If it didn't have that, it wouldn't be able to get in. And so you see these, when populations get really abundant, there's probably only a very limited number of viruses that infect that organism, and then they will take that population.
Why over time wouldn't you see? Why over time wouldn't you see the plankton that are being attacked evolve defenses against the virus and get to a state where the virus doesn't, cause such huge turnover in the organism?
Well, in fact, this is happening all the time. It's a continuous. We could think about it as a war, but there's something in the Alice in Wonderland, which we have in ecology, called the Red Queen, which is, I have to run as fast as I can just to stay in the same place. And that's what happens in biology. And so you find that these viruses essentially chase the hosts that they infect, and the hosts are continually adapting different mechanisms to kind of fight off the viruses. But we have to also understand that there's many, many different types of viruses, and they're all doing different things. So if we look again at humans, there's probably at least 100 different virus diseases in humans, which are caused by different viruses. And so just because you manage to get away from one doesn't mean there isn't another one. And whatever adaptation that you may have acquired, which allowed you to escape infection from one virus, may make you susceptible to infection by another virus. And there's actually a number of examples of that. So for example, if say a virus will often recognize a highly conserved protein because over evolution, that's very hard for the organism to mutate, because it's absolutely dependent on it. And so there's examples of proteins that have specific mutations, which means the virus can't infect it anymore, but then that protein doesn't do its job as well. And so there's a disadvantage to that mutation because the protein, say, doesn't transport a specific nutrient as quickly as it could otherwise. And so when the virus pressure is gone, all of a sudden the virus can't infect anymore. It's diminished. The risk of viral infection goes very, very low. Then there's an evolutionary pressure to mutate back to the original form again, because that's the one that's effective, right? And so you have these kind of flip-flops back and forth, and it's very, very complicated. Almost anything you can think of, viruses and hosts have kind of figured out a way to interact with each other. And some of them are amazing. I mean, if we look at our own genome, about 8% of our own genome, the human genome, is actually made up of viruses, which have
I've heard that many times, yeah.
Yeah, so where they get
Amazing.
locked into our own DNA and they can't get out.
What are some of the dynamics of viruses attacking plankton? I mean, are there ones that attack only when plankton are in a bloom state or in a biofilm state, and others that attack when they're solo? Any observation there?
Yeah, so again, typically we kind of go back to what a virus is, and I think I'm just going to kind of roll back here a little bit because it's kind of fun to think about it. And I don't think about these things very often either. And I was giving a talk one time to a general audience of potential donors for an institute for a after dinner talk. I was trying to figure out, well, how do I communicate what a virus is and the size of them? And I hadn't really thought about it before, but they're incredibly small. So for example, if you take a virus and you scaled it to the size of the head of a pin, for example, so you could see it, and I was scaled by the same amount, I would be 150 kilometers tall, right? So they're tiny. And yet they're so abundant that in every milliliter of seawater on average, there's about 10 million virus particles. So every time you go swimming, you swallow about as many viruses as there are people in North America, just from the amount of water that you take in your mouth, right? And yet if you were to take the viruses in the ocean and stretch them end to end, they're so abundant that they would go 10 to the 7th light years or further than the nearest 60 galaxies. So they're there in this incredibly high concentration. In seawater, they have very short lifespans overall, so they turn over very, very quickly, and they're very, very host specific. So to come back to your question about are there some which only infect solo organisms? Well, the way a virus makes a living typically is it has to find that specific host, and because their lifespan isn't very long, when cells are in low abundance, then in fact the probability of actually infecting them is very small. So some viruses have actually got around that problem because they just, when they find a host cell, they just integrate into the DNA of that host and they just kind of hang out, right? Or they integrate in with the cell in some sort of way, maybe as an extra piece of DNA. So every time the host replicates, the virus replicates with the cell until times are great. And then when times are great and that cell starts to grow really fast and becomes abundant, then the virus goes into this lifestyle where it kills the host and produces many, many viruses. And you can think over evolutionary time why that would work. And it works because if an organism is growing really, really fast, then probably there's other organisms out there just like it that this virus is going to be able to find. But when things are not going very well, then it's probably the chance of finding a new host are relatively small. So why don't I just hang out inside this host cell that can look after me so it can repair my DNA? I can just replicate alongside with it, things like that. So there's there's these different sorts of lifestyles that viruses adapt, and not every virus can do everything. So there's many
some viruses can get inside let's say a plankton or a cell and sit there and wait till the cell fattens up and then eat it, or then slaughter it, essentially like fattening up a pig. but instead of from the outside, you're on the inside, and they'll
Yeah,
be commensal, and then they'll all of a sudden lyse the cell when they're ready.
right. And because these are single-celled organisms in the ocean for the phytoplankton and the bacteria plankton that are out there, when the cell's growing slowly, it just, every time the cell divides, the virus probably divides as well, has another copy, it goes along the host. And you can imagine that particular host cell is very resource limited. It doesn't have enough nutrients and things like that. And in a way, the virus is reading that. And so it recognizes that it maybe not even have enough resources to produce, a bunch of virus cells to be released. So yeah, it's an interesting dynamic. And that's not every virus, only some viruses. There's this huge diversity. So viruses, again, they are able to encode their genetic information using, single-stranded RNA, double-stranded RNA, single-stranded DNA, double-stranded DNA, all of these different ways of making a living, whereas all cellular life just uses double-stranded DNA. So again, all of these viruses have different lifestyles and how they interact with the host organism.
So you could theoretically have a virus or viruses that live inside of phytoplankton, and then, for instance, when they bloom, certain genes are activated or certain signals are created inside the cells that say to the virus, all right, it's time. And then maybe they wipe out the population.
Exactly. You're right. Exactly. So they're increasing they're still growing in a sense, because every time those cells divide, they're producing more viruses inside the cell, but not very many. And then when the population becomes very large, potentially those viruses could release and cause lysis of that particular population. But that's one life. And there's many of these things that we just don't know very much about, right? These are become the ones which are integrated or become part of the, as a host, especially if they're just a piece of DNA inside the host as opposed to integrating into a genome, become much more difficult to study because it's a separate piece of nucleic acid. And so if you're doing sequencing or things like that, you always wonder, well, maybe it's just a contaminant because it's not actually integrated into the genome. So it's these things are kind of difficult to study and understand how they're regulated. Some are well understood, but many are not.
So which viruses are you studying in particular, and what functions are you looking after?
Oh, wow. I mean, we're so plastic in terms of what we do. It's funny when years and years ago, when I put in my first grant proposal to the National Science Foundation, they essentially wrote back and said, well, we have nobody actually to review this because there's nobody that works with viruses. And ultimately, they sent it half to virologists and half to oceanographers, both of whom hated it. The virologists only study one virus. The oceanographers said, well, we're not really interested in viruses, we're interested in process. And so over the years, we've studied so many different kinds of viruses. I say a lot of the work that we do is viral discovery work. We don't know what we're going to find. And so that's why we've ended up discovering so many types of different viruses. And we've had funding, in the past from NASA and the Canadian Space Agency. How do you look for life if you don't know what you're looking for? Because again, you find these different kinds of viruses of stories. So, a virus again is an obligate pathogen, which can only replicate by finding a cell and infecting it. And yet, one thing that we came across a number of years ago was this is a little, again, single-celled zooplankton. So something that would eat bacteria plankton or small phytoplankton, just a little organism that has a flagellum on. And we found what's called a giant virus that infects it. And so these giant viruses have very, very large genomes for a virus. So much. And so this was, at that time, the largest one of these viruses had ever been found that infected a marine organism. And we were having a lot of trouble growing it. In fact, we had isolated this virus many years ago, and I had so many students and postdoctoral associates working on it, and they all just gave up because it seemed to go really well for a while and then would kind of fall apart. And it turns out what was happening is there was actually something called a virophage. So it was a small virus which parasitized the giant virus and actually killed the giant virus and ultimately saved the little zooplankton. And this became, in a way, is another way of adaptive immunity. And so when we sequenced the little virus, we were really surprised to see that it didn't look like viruses at all. It actually looked like transposable elements, which are found in other eukaryotic cells. And so the PhD student, Matthias Fischer, who did this work, is now at the Max Planck Institute in Germany. He took the system with him when he went there to continue working on it. And he's now discovered that these little, what we call virophages, jump in and out of the genome of the host and actually give it immunity to infection virus.
Wait a minute. The virophage material was part of the DNA of the host and was activated to fight a virus that was attacking the host?
No, it's actually a little virus. It's actually its own virus, but it's absolutely dependent on the replication of the giant virus in order to reproduce itself. And it has these the things which start the replication of the giant virus. There's a certain sequence of DNA which the small virus recognizes, and it also causes its own replication. So as the giant virus replicates, so does the little virus, and then it actually ends up killing the replication of the giant virus. But this
happens inside of the phytoplankton cell, or?
Yeah, inside. In this case, it's not a phytoplankton, but it probably happens in phytoplankton cells as well. We just haven't isolated. But yes, in this case, a single cell zooplankton. But now there's many of these things in the ocean. They're just really, really hard to work with because they have to replicate on a cell which is being infected by a virus. And so we know that there's lots of these little viruses in the ocean that have this lifestyle. But unless you have a host organism that's infected by a giant virus, then you can't isolate the little virus. And then the little virus ultimately kills the giant virus. And so then it leads to its own demise as well, because it requires infection by the giant in order for itself to survive. But now Matthias has found that at least in some cases, these little viruses can also integrate into the protists, the little microzooplankton, and hang out there and replicate along with the host cell as well. So in case it ever becomes affected by a giant. So it's just that these patterns of viruses and what they do and how they interact and how they drive evolution and how they drive community structure and biogeochemical cycling, all of these things are very tightly entwined.
How do you think viruses find their targets? If they're virions and they're supposed to be inactive and they can't move on their own, and they're so small, in such a vast expanse, how do they ever find their targets?
Yeah, so it's the random walk is in most cases. That's what seems to drive most of these interactions. But there's also some other really interesting things that are going on. For example, there's one virus which infects a phytoplankton, which is a symbiont in a protist, a heterotrophic protist. So it's an alga which only lives inside which would be a zooplankton, I guess, right? And so that's what this virus infects. And so in some cases, there's an example which is cool that the virus seems to produce a giant coating of sugar on the outside. And that's probably to encourage this little zooplankton to eat it so it can infect the host cells, which are only found inside. So it's really there's sort of almost anything that you can imagine. Evolution has come with something that can kind of figure out what's some way of getting around the problem, right? And so, it's intriguing.
Doesn't this seem to say that viruses are alive, at least once they're inside of a cell, because of all the contingent things that they can do and the evaluation of information?
That's an absolutely great question, and one that's there's lots of discussion about. And so there's a way of thinking about this, which I think is a really kind of interesting perspective. Someone who's really been promoting this is a fellow called Patrick Fortier, who's at Pasteur in France. And but the idea is that the virus particle is just a dispersal phase, so it's not the active virus, right? And so what I always tell my students is you walk into a room and you don't look into the classroom and say, I can't believe it, there's 20 billion people in this. And that's because we don't count the sperm and we don't count the eggs of everybody that's sitting there, even though they contain all of the genetic information to be a human. So the phenotype we recognize as being a human is the person that's sitting in the chair. And the same way that we go to the ocean and we don't say, God, there's 20 billion fish out here, because we don't count the fish eggs that are floating in the ocean. We don't call them fish, even though they have all the genetic information of fish. Or you could go on and on with the analogies with plant seeds and things like this, right? And so the thought is that the virus is just the dispersal phase, right? So that's the equivalent of the, maybe not sperm and egg, which are reproductive, but seeds, for example, from plants. So it's a dispersal phase. The actual phenotype of a virus is actually the infected cell. That's the virus, because at that point, that cell's sole purpose is just to produce viruses, right? So the living virus is actually the infected cell. And the virus particles, which are so abundant and widely dispersed, are in fact just the dispersal phase. So I would say, yes, absolutely, viruses are alive.
Oh, that's good. Do you study viruses in microscopic, fish, whales, dolphins, things like that? Does anyone do that?
Yeah,
I would think they would have very interesting stuff there.
No, it's a good question again, because we just had some papers out on viral discovery in salmon, and maybe not surprisingly, we've discovered all kinds of new viruses there. So working with colleagues of the Department of Fisheries and Oceans Canada, they had this kind of unusual problem in that they have a high throughput technology, which essentially is able to rapidly screen a fish for all known pathogens of fish. And essentially they can do 96 different pathogens against fish. And so in this case, when there were dead and dying fish, which had obvious pathology, and they put them against their high throughput array, and they found no pathogens that were known, then they would hand over all the sequencing to us and see if we could find new viruses. In fact, so a postdoctoral associate that was working with me, Gideon Mordecai, went through all this material and found that all of these new viruses, which were absolutely not known in fish, or at least certainly not known in salmon, and in some cases weren't really known in fish at all, right? And so so that would be one example of where we've looked at viruses in sort of macroscopic organisms. So so there's lots of kinds of areas of viral discovery. Basically, we know so little about the viruses in the planet, something called the virosphere, which is, the virosphere is all those places on Earth that viruses exist and are affected by viruses, which are essentially everywhere that cellular life is found. And even we had a paper a little while ago, 2 years ago now, looking at the viruses above the planetary boundary layer. And it turns out that even if you go up 9,000 feet in the atmosphere, the vertical flux of viruses, just falling out of the sky, is more than 1 billion per square meter per day. There's just massive amounts of viruses.
What kind of viruses will be up in the atmosphere?
Well, it's really interesting. They get swept up from the surface of the Earth, and so we could actually see where the viruses came from the desert or from the ocean. So this was a collaboration with Professor Isabel Richet at the University of Granada in Spain, and she had these collectors on the top of the Sierra Nevada mountain in Spain. And so we had a conversation one day. So why don't we look and see if the viruses are in there, right? You're collecting all this material, and so indeed it turns out that there's a massive number of viruses up there, and once they're that high in the atmosphere, so the planetary boundary layer is essentially above the area where there's much friction in the Earth. So they're up in that part of the atmosphere where they're free to travel around the globe. So once they're up there, they can get to North America. They can get anywhere on Earth, right? Just by the currents, the winds, currents, the atmospheric wind. Yeah, so we can actually see whether the viruses came from the ocean or whether they came from Sahara Desert, depending on just the wind patterns.
So viruses, I guess because they're so small, anything could carry them. But so I guess I guess there's viral cycling throughout the entire globe because of the winds.
Yeah, and the thing that there are viruses circulating around the globe, and this was one of the things that drove us to look in this direction as we were doing some genomic studies on specific kinds of viruses and sequencing them. And what we found was the identical viruses in lakes in Germany, in meltwater ponds, the Arctic ice shelf, in the middle of the Gulf of Mexico, and catfish production ponds. So we said, viruses are supposed to be host specific. So it's very unlikely that there's going to be the same virus or the same host organism growing in Lake Constance in Germany at 2.5 kilometers deep in the Arctic Ocean in the Gulf of Mexico. And so the hypothesis was that these viruses must be being widely dispersed. And so in fact, we sort of think about viruses as not being dispersal. So they're free to disperse very, very widely. Now, that if we think about connotations such as viruses infect mammals and things like that, well, they tend to have pretty limited dispersal because they're highly specialized in terms of infection process that they need to infect them. So in most cases, those would not be dispersed widely, but there are some viruses that are pretty recalcitrant, potentially could be dispersed far, not coronavirus.
Yeah, that's crazy. When you observe bacteria in the ocean, let's say, or other creatures, phytoplankton, et cetera, we talked a lot about viruses infecting them and endogenizing or just living with them and lysing them, and et cetera. Is there any use of them from the other side? Do bacteria or other creatures seem to deliberately use viruses to their own ends? Or are they just preyed upon by them, defended, they defend themselves and that's it? But do you see any, I don't know, use them as nutrients?
No,
do they do anything
question.
like that?
Fantastic question about whether, let's look the other side. What benefit do viruses have? And in fact, this is what I preach about most commonly, is that viruses are mostly our friends, not our enemies. And so, for example, if you look at the microzooplankton in the ocean, it turns out that if you look at the number of virus particles that they eat, that is sufficient to provide all of their nitrogen, say phosphorus requirements. They don't need anything else, right? So because viruses are incredibly rich in phosphorus proteins that are surrounding nucleic acids, so they can be a food source. There's also some really intriguing things because viruses become integrated into the DNA of the host. And there was one study years ago by a colleague, which I thought was just amazing. He was studying bacteria in this case, and he was looking at viruses and how fast they're damaged by ultraviolet radiation. And so he was doing that by exposing the viruses to ultraviolet radiation and letting them infect the host. And this is a fellow called Tyler Cockjohn. And what he found was that when UV radiated the host, of course, viruses are badly damaged because it damages DNA. And so, but what he found is he exposed the viruses to ultraviolet radiation. They were able to, the ones that could still infect the host ended up expressing genes that caused the host to produce a dark brown. And so in this case, the virus was inside the host cell where it was somewhat protected because the host cell could repair the viral DNA, but it was also giving the host cell tools so that it could avoid damage by the ultraviolet. This is an area that we know very, very little about, and that's viral mutation. So I think that probably most viruses that we have a close association with actually help the organism in some sort of way. And there's some nice studies.
They're obligate symbionts then, not just pathogens.
So they're mutualists. They help each other out. Yeah. So Forrest Rower at San Diego State University, his group had a really nice study in which they argued that certain viruses that we inhale, for example, because we're completely surrounded by viruses, we're inhaling thousands of them all the time, will get into our lungs. And actually their little heads get stuck in the mucus and they put their feet outwards. And so that means all the bacteria that we inhale that virus can infect, the virus infects it immediately and kills the bacteria. So in other ways, it's our first line of defense against some bacterial pathogen. So that's a real cool story. And then we look at our own nucleic acid, our strong evolutionary relationship. And so, for example, viruses, so the placenta of mammals, for example, one of the proteins involved in the formation of placenta of mammals, protein that was donated by a virus, one of our ancestors hundreds of millions of years ago, right? 100 million years ago or so, probably. And so we have this, if it weren't for those viruses, there would be no mammal. Same with our nervous system, some of the major proteins in our nervous system.
Huh. So, well, this is kind of a weird question, but you know how food can be fermented by bacteria, and pretty much everything we eat is worked on by bacteria. Is there such a thing as like a viral fermentation where, a food while it's alive is lysed and kind of chewed up or acted upon by viruses and then consumed by another creature? Can viruses like quote unquote ferment food and change it so it's amenable for something to eat it when it otherwise wasn't before?
Yeah, I don't know the answer to that question. I mean, the viruses, the more we learn about them, the more we realize that they're encoding all kinds of functional genes that do things we don't really know why they have those. And mostly they're genes which at some point have been co-opted from organisms that they infect. And then they've been worked upon to be viral genes. And so, there's no reason, for example, why a virus couldn't encode certain genes that would allow maybe an organism to either give it traits that it could carry out other biochemical functions, or maybe damage it in certain ways. In other words, there's probably, if it increases the viral fitness somehow and is able to allow the virus to survive better. So in other words, by modifying a cell so it becomes infected by something and gives it a different lifestyle, then so be it, it probably does. I mean, one really cool example, it's not exactly like that, but is cholera, right? So cholera is caused by a bacterium called Vibrio cholera, but almost all Vibrio cholera are non-pathogenic to humans. In order to become pathogenic to humans, it has to be infected by two different viruses. And those two different viruses causes cholera to be a human pathogen. And then in that case, cholera can invade humans, cause huge amounts of diarrhea, which of course is very beneficial for the bacterium because it has lots and lots of substrate to grow on, and is also super beneficial to the virus because it produces tons of viruses. So certainly viruses can moderate the hosts that they, and I can think of other cases of pathogens in general, right? So not necessarily just viral pathogens where they modify the behavior of hosts. So for example, making a caterpillar more attractive to birds. So the birds will eat the caterpillar so the parasite can replicate inside the bird, right? So there's sort of traditional things that parasites do everywhere.
Yeah, it's just amazing. So what are there any big concepts that you sense that you're getting close to understanding with your research?
Yeah, one of the things that we're really kind of focused on now is something that we call taxon-specific mortality, right? So almost all the numbers that we have for looking at viral infection in the ocean, and we look at the microbes in there. So you can think about it, there's thousands of different microbes, right? And there's tens of thousands of different viruses infecting these thousands of different microbes. And so as an ecologist or as somebody that's a biogeochemist, you really want to be able to answer some of those questions you asked me about, well, what are the viruses infecting? What species are they infecting at what time? And so we've come up with some new tools, for example, to try and unravel that problem, to look at taxon-specific lysis, where we can look at we can look at an unlimited different number of taxa and look at what their mortality rates are as a result of lysis. So that's something I think is really cool, because then we can start to, so that's really cool if we can start looking at taxon-specific lysis, because that'll give us some sort of handle in terms of how viruses are regulating population. So that, I think we're making good progress there. Some of these virus pathogens of fish, I think they're going to be really important in their impact on different organisms. And then I really would want to try and expand that into other areas where we can really start to understand the role of viruses in regulating natural populations. So we really know, the only thing we know viruses for basically are things that are either really economically important, right? Or maybe hugely ecologically important, or maybe they infect, we know a little bit about things that infect charismatic megafauna, right? So we know a bit about seal viruses and whale viruses. People think they're cool and they're probably ones, but most of the life in the sea, we have no idea what virus is.
Well, very good. Well, Curtis, we're just about out of time. This has been a really cool call. What's the best way for people to learn more about your research?
I guess what everybody does right now is just Google. So we publish in a variety of different areas and a variety of different platforms. And if people are interested in learning more about viruses, and I think the main point that I really like to get across is that even despite this horrible thing that's going on right now with the pandemic, is we actually our evolutionary existence depends on the fact that we're closely associated with virus. The health of the planet depends on the fact that viruses are doing a lot of the heavy lifting that allows nutrients to cycle, organisms to survive, populations to change. Because if there's no mortality, then there's no life, right? Because you have to be able to repurpose those nutrients and things like that. So I think if people start thinking about viruses the way we think about most things, that they're actually part of our evolutionary existence, then it helps to frame these things in a clearer way.
Yeah, very true. Well, Curtis, thanks for coming. It's been, like I said, a really great call. I appreciate it.
Yeah, great to talk to you, and good luck with putting this all together. Take care.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.